Since the dawn of our species, our brain has been playing a concert that no ear has ever truly heard. A symphony of molecular signals, invisible, silent, yet powerful enough to ignite thoughts, love, and madness. But how can we record and understand a coherent score of this complex brain activity? It's one of science's great mysteries. Every brain cell plays according to a score, an internal blueprint that we cannot fully decipher. But what if we could solve this puzzle and understand how our brain works? The implications would be profound. We could decode the patterns behind our thoughts, map the roots of diseases like depression or Alzheimer's, and perhaps even glimpse the very nature of consciousness itself.
Of course, we want to understand how the brain fundamentally works. And its function depends on around 100 billion cells communicating with each other through various types of messenger molecules, so-called neurotransmitters. Perhaps imagine it like this. These neurotransmitters are like specific notes in a piece of music. A C, a D, an E. We know they're in the brain score, but we often don't know when exactly are they played, how long do they resonate, and which cellular instruments actually bring them to life. It's a complex symphony across these billions of cells, and that's precisely where we come in. We want to track when and where these important notes sound in our head to better decipher this score of the brain.
To decipher the complex communication in the brain, its multi-layered score of cellular instructions, science developed impressive tools. They help to identify molecular signals, the notes in the system, and also the cellular mechanisms by which these notes are played, the instruments of the brain. But to grasp the entire interplay, the full symphony of this vast brain orchestra, more is needed.
So to understand why we're doing something new, it might be good if we briefly look at the limitations of current techniques.
A crucial breakthrough came in the early 20th century with fluorescence microscopy. Here, special markers, fluorescent molecules that specifically attach to certain molecules in cells, make them glow under the microscope by absorbing light of one color and re-emitting it in another. These marked molecules and the cellular structures they belong to are crucial for the cell's function, for its music. We can think of them as the instruments with which the cell operates.
You see, it's like with this picture here. The details of the setup, they're perfectly clear, but it remains a still image. What the cells exactly do, that's just missing here.
But science wanted to observe the musicians, the cells themselves, and the processes within them live. However, a deep look directly into us humans with this light-based method is another challenge. Our skin and dense tissue simply block too much light.
With these binoculars, I can see a small group of cells, or musicians, as they play live and in great detail. But the entire orchestra of the brain, the vast interplay of all the cells, that I just really cannot see with this approach.
For the overall view of brain activity in living mammals, another method, familiar to many, came into play.
That's where fMRI comes in, functional magnetic resonance imaging.
The basic idea, active brain cells need more blood, like muscles. In the early 90s, fMRI made the breakthrough of measuring this blood flow non-invasively, allowing us to watch the brain think. This famous bold signal is a strong indicator of where nerve cells, the musicians of the brain, are currently particularly active.
So fMRI shows us where in the orchestra there's a lot going on, where it's buzzing with activity, where there are many cells simultaneously firing. But the individual notes, the specific messengers and molecular mechanisms, they remain hidden. But what if we could somehow bring these two worlds together? If we could, let's say, combine the molecular precision of fluorescence, which enables us to hear the specific notes of the messenger molecules, with the reach of fMRI, to then capture these signals live throughout the entire brain.
This is precisely the challenge Dr. Robert Ohlendorf and his team at the Max Planck Institute for Biological Cybernetics in Tübingen are tackling. Their approach, to equip the fMRI with molecular prompters that indicate which specific notes are currently being played in the brain.
Well, the basic idea was actually to make fMRI a bit smarter. Normally, this bold signal only shows us quite non-specifically that there is activity somewhere. So we wanted to find a way to teach fMRI to tell us which specific messenger molecules are involved in this activity. This is which node the brain cells are playing. And for that, we are developing special proteins as tiny sensors.
These miniature sensors, co-developed by Dr. Ohlendorf and his colleagues, are named avatars, molecular spies, so to speak. Their blueprint is transported into brain cells, which then produce these spies themselves. Each avatar is calibrated to hear a specific note, a specific messenger substance. When the avatar recognizes and binds to this messenger, a molecular switch is flipped inside it. This switch, in turn, signals the muscle cells of nearby blood vessels. Relax. The vessels dilate and more blood can flow.
And precisely this local dilation of blood vessels then creates an artificial change in blood flow. The fMRI scanner measures the signal very similarly to natural bold signals caused by normal brain activity. But the key is, because we know the properties and signature of our avatar signal very precisely, we know, for example, our avatar only reacts to dopamine, we can distinguish this artificially generated signal quite clearly from other brain activity.
With avatars, the general hum of fMRI thus becomes understandable music. Rather than simply hearing that the orchestra is playing, we can now clearly identify the individual instruments, these specific messenger substances, and better understand their role in the symphony of the brain. With avatars, researchers can thus listen live to the specific notes of messenger substances in the entire concert hall of the brain. This makes answering old questions and posing entirely new, precise ones a tangible possibility.
Yes, that's exactly what drives us. The avatar technology now gives us tools to investigate truly fundamental mechanisms of the brain. Questions like, how do we actually learn? How do emotions arise on the level of messenger molecules? Or even, what distinguishes human and artificial intelligence when you look at the underlying mechanisms? And of course, the great hope is that this deeper understanding will also help us to better comprehend the molecular causes of serious diseases. For example, Parkinson's or Alzheimer's. That would be a very important step for future therapies.
As researchers learn to visualize and understand the fine molecular imbalances in the living brain, this basic research paves the way for targeted therapies with significantly fewer side effects, a real chance for future personalized medicine.
Sure, such prospects really speak to the imagination, but one must also say the direct application of this specific avatar technology in humans, well, for such scenarios, that's very much in the realm of science fiction today. But let's just hypothetically play with where such deep understanding of brain chemistry, where it might potentially lead us.
Imagine a not-too-distant future. You feel down, you go to your doctor. Instead of a long questionnaire and the usual pharmaceutical trial and error, you are slid into a special scanner. Invisible, yet highly sensitive, molecular sensors in your brain map your brain chemistry live. The result? A precise mood profile. Your doctor sees immediately, aha, a slight serotonin deficiency in brain region A combined with a slight overactivity of messenger substance B in network C. You then receive a medication tailored exactly to your individual neurochemical imbalance, a subtle correction that restores your ideal state with minimal side effects.
Yes, that would be the dream, of course. And the potential, it's massive. That's why we're really excited to work on this. But it's a long road to get there. We really have to keep working step by step. And our next step now is to, well, you could say, properly train these avatars. What we do in the lab is to generate a huge variety of slightly different sensor variants. These can be millions of candidates, and they then have to undergo a pretty rigorous testing procedure in the cell culture, like a stepwise tournament. Imagine it like that. Only the very best that specifically recognize the messenger molecule and also give a strong, clear signal advance to the next round and are then further optimized.
A long road, yet the avatar technology from Dr. Ohlendorf and his team promises a new era of understanding.
Yes, our clear goal is to use these avatars to decipher the language of the different messenger molecules in the brain, so that we can better understand these complex melodies behind our existence.